Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about meiosis and trying to figure out how it is different from mitosis, what it's used for, and all kinds of good stuff. So just to kind of give us a little bit of basic vocabulary on heredity, we've got some things we need to just run through real quick. Genes are units of heredity that are found on DNA, and the locus of a gene is its specific location along a chromosome. Gametes are specialized reproductive cells found in animals and plants, and they're going to help transmit genes from one generation to the next. And there are kind of two different types of reproduction. We've got asexual reproduction, which is where a single individual is the sole parent and passes down all of its genes to the offspring, which are also sometimes called clones. And then we've got sexual reproduction, where we've got two parents, each contributing an equal portion of their genes to help create genetically diverse offspring. We talk about asexual reproduction. That's usually going to be some form of mitosis that's occurring, whereas sexual reproduction is going to be utilizing the process of meiosis. Now, sexual reproduction and meiosis are different things. Meiosis is used to help facilitate sexual reproduction. So some other vocab words that we need. Karyotypes. This is a display of all the chromosomes found in a human cell. Human cells have 46 chromosomes, 23 pairs. We've got homologous chromosomes, which are the pairs of chromosomes that have the same genes. Remember that homo means the same, logous means information, so homologous or homologous, same information on the two chromosomes. You've got your autosomes, which are the pairs 1 through 22, and then you have your sex chromosomes, which are the X and Y chromosomes that help determine sex. Lots of organisms have different ways of determining sex, but they are usually, there is some sort of sex chromosome that helps to differentiate. How exactly those look is obviously different from species to species. We have diploid cells, which are your two-end cells, which are also sometimes known as somatic cells. Those are your body cells. And then you have haploid cells, which are gametes, and they are so 2N means that you've got a full set of chromosomes. You've got the full 46, whereas your haploid cells only have 23. They have basically a half of the full set. So human life cycle. Human life cycle starts off with the sperm and the egg. The sperm and the egg are both haploid gametes. They're going to fuse together in the process of fertilization. Once that fertilization occurs, you now take the 23 chromosomes in each of them, put them together, and now you have the diploid zygote. The zygote is going to go through mitosis to grow and develop. It turns into, an adult human. And then the germ cells, which are the ones that create the sex cells, are going to create new sperm and egg cells, which are then going to fuse, form a new zygote, and then the cycle continues. So we spend the vast majority of our lives as diploid organisms, but not all organisms use the exact same trend. In fact, there are some organisms that spend most of their life as haploid organisms. So animals, most animals, again, have very small, short haploid generations, and then they go into a diploid state. Plants and algae spend most of their lives as diploid organisms. However, they also have haploid parts of their cells that are, that they spend a lot of time in. Things like mosses and stuff spend the vast majority of their lives as haploids and only form diploid organisms for a small period of time. Things like fungi and protists spend most of their lives as haploids. They then combine with other haploid parts, other haploid individuals in order to form diploid organisms that then go through meiosis to then produce the part of the cell of the organism that actually does most of its life functions. So it's kind of interesting how you have these variations in whether or not you're spending most of your life as either a haploid or a diploid. So there's no real advantage to one or the other. All right, let's talk about meiosis, right? Producing these haploid cells. Basically, the idea is that we're going to start off with a diploid cell. We are then going to half the number of chromosomes in order to get to a haploid cell. And we're going to do this in two separate cell divisions, which are known as meiosis I and meiosis II. So here's your first kind of difference between mitosis and meiosis. Mitosis occurs in one cell division, whereas meiosis occurs in two cell divisions. So the first phase of meiosis, meiosis 1, begins just like mitosis with prophase 1. Here you've got your chromosomes, again, condensing, and the homologs are going to be aligning. So the homologs are your two pairs of homologous chromosomes, one of them from the mother, one of them from the father. They are going to be lining up with one another. So you basically have 4 whole chromosomes that are lining up together, and they're going to actually connect to one another in what we call synapsis. And this is where crossing over is going to occur. The segments of the DNA are swapped between the pairs. So basically what we're doing is we're creating chromosomes that are no longer just the information from your father and just the information from your mother. They're actually now a mixture of information from both your mother and your father on a single chromosome. This is going to be one of the ways that we're going to help to increase genetic diversity. The actual place where the crossing over occurs is called a chiasmata. And then the chromosomes are going to begin to move kind of into the metaphase. Here the homologous pairs are arranged along the metaphase plate. So what's going to happen is that we're going to get one of your, or we're going to get the pair from your mother and then the pair from your father that are going to line up, and they're going to line up randomly along the metaphase plate. Each of the homologs is attached to a microtubule at the kinetic core, just like we saw with mitosis. Anaphase 1, the homologous pairs are going to be separated. They're going to be pulled apart from one another. The centrosomes are not broken apart. So the sister chromatids remain attached, which again is a difference here between mitosis and meiosis. The sister chromatids are not pulling apart during the first cell division. Then we go through the same thing of telophase and cytokinesis. We form 2 new chromosomes. At this point, these are sometimes, called haploid. Sometimes they're called diploid. Frankly, it doesn't really matter. You've got 46 chromosomes in there, and that's all you really need to know. It's just a halfway point, kind of along the way. Now we go into the second stage of cell division. We've got prophase 2 happening. Again, everything kind of breaks down. The spindle apparatus forms. We don't have any crossing over that occurs at this point, though. Crossing over only occurs during prophase 1. Metaphase 2. The chromosomes are going to position along the metaphase plate just like they do in mitosis. So it's the same kind of lining up. We've got the centrosome that lines up right along the middle, and then sister chromatids are going to be pulled apart like they are in mitosis, moving to opposite ends of the cell. And then the sister chromatids are going to do the same thing that they do. They're going to decondense, form back into the chromatin, and reform your new daughter cells. Except now at this point, we have 4 daughter cells that are haploid, and they are all genetically different from the parents because of the crossing over that occurred and because of the lining up of them along the metaphase plate. So there are 3 different ways that we get the majority of our genetic variation when we go through the process of both meiosis and sexual reproduction. We have independent assortment, which is the fact that each gamete has a 50% chance of inheriting the paternal or maternal chromosome from a set of homologs. So when they line up along that metaphase plate, you have a 50% chance of either having the paternal or the maternal. So that's going to increase variation. You've got crossing over, which is going to result in a huge different number of variations, and just in terms of which genes get swapped over from one chromosome to the other, and just how many times it occurs, and lots of different ways that can help to increase variation. And then finally, you've got random fertilization. Each gamete itself represents a 1 in 8.4 million possible chromosome combinations, excluding crossing over, which leads to a 1 in 70 trillion possible different zygote combinations, just in terms of whether or not the chromosomes are lining up, not even taking into account crossing over. So lots of different sources of genetic variation, and the way that we can have such a wide variety of people, and you don't really ever have 2 people that look the same. You can imagine why there hasn't even been 70 trillion people born yet, so that have lived on the earth. So we haven't even gone through all the potential possible zygote combinations. Crossing over looks something like this. Remember, again, it occurs during prophase 1. The non-sister chromatids are held together during synapsis. These are the ones from the mother and the ones from the father. We got the chiasmata that are going to be forming where crossing over is going to occur. We now end up with chromosomes that basically have swapped segments. So you have hybrid chromosomes that are partially maternal, partially paternal, and we end up with recombinant chromosomes, and we get distinct possibilities in each of the daughter cells. Independent assortment, we can either have the paternal chromosomes lining up on the same side, or we can get them flopped over so that now you've got the paternal and the maternal on both sides, leading to different combinations of the chromosomes in the daughter cells. And so you can quickly see how we can arrive at this large number of possible combinations in the offspring. Now, you might be wondering, why is it so important that we get all of this independent assortment and the crossing over? Why is it important that we have all this genetic diversity? That's very important for evolution, right? We need to have a large variety in the offspring, so that way we can be better adapted to certain situations, survival of the fittest, all that kind of thing. We'll talk about that more when we actually get to our evolution unit, but this is the context for why it is advantageous to reproduce sexually. So, thanks for watching, and I will see you all next time.